Compiled a big overview deck for you guys. I've already got the accent question, is this posted on Canvas? The answer is I don't post my review decks on Canvas because they're mostly comprised of your own slides from these decks, and they're put in sequential order, so as you guys are, as we're moving through this, you can just follow in order with your own decks. A whole bunch of caveats to get out of the way though. I've been doing it this way for a while. As I've said previously, I don't like spoon feeding questions on the exams or anything like this. So this is not like a blueprint to the exam, and I don't deliberately break up the silhouette of the exam either, but I think it ends up pretty well broken up. The intent is just to kind of go over some things that I think need extra emphasis, and generally the feedback I get is that it ends up being pretty helpful to you guys. This time is a little bit different in that I did add some extra slides. It's not testable material, but it's just a couple like exam style multiple choice format questions just for you guys to practice on. Obviously it won't be on the exam itself, but that's feedback I often get is why don't you throw on some more MCQs. So I did do that with a couple of those, and they're harder than what you would see on the exam on Friday. So it'd be good for us to cut our teeth on something a little bit harder. In terms of abbreviations, a couple other things on this exam. There's a lot of abbreviations in immunology. On the exam, I'm not going to throw around a lot of shorthand or these colloquialisms or anything like that. For example, if a question is about natural killer cells, I will write out natural killer cells or natural killer parentheses NK cells. So the point of these exams is to be as clear as possible, not to confuse you, befuddle you, or give you anything else to siphon away brain power. Just be as really straightforward. The one exception might be, well, a couple of exceptions, like if I've defined APCs, which are, what are APCs? Great. So if I've defined that a couple times, I might later on in the exam just start calling them APCs. And then you can also, if you forget what that is, you can refer back up. MHC currently is just MHC on the exam. So you kind of got to know what that is. It's in the decks, and hopefully you do know what that is. Do you? Histocompatibility complex. Yes, exactly. It's just a lot to spell out in the exam. So like that currently is in its abbreviated form, and spoken, hopefully you're used to hearing like MHC1 and MHC2. So, yeah.
What about like B7 versus CDAB?
Oh, good question. Good question. CD, I will reflex the CD. There's a mock question with B7 on here, but that's a mock question, it's not on the exam. Yeah, some of these do have two terms, but I try to use the one that I've like really been using on the slides and in lecture a lot. Yeah. In terms of pictures, I've stripped away a lot of the pictures on this exam, although a lot of my exams do have pictures. I just felt like most of them were unnecessary. So it always helps to look at those pictures and understand, like, those cartoons of like what a macrophage looks like, for example. Antibody structure, know that. If you were given like a cutout of a cell wall or a cell membrane and asked with a bunch of numbers pointing to different parts of those structures and asked to identify like what the phospholipid bilayer is, on those structures, that's fair game. But yeah, this exam I didn't really put a lot of pictures in it, so a lot of it's textier than my usual exams. I think those are all the caveats. If I think of any more along the way, I'll let you guys know. But yeah, keep your questions coming. Just ask me what's in the exam. Okay, so we'll start off with the first deck. There was a bunch of intro stuff in there. I always encourage you, of course, to go over that stuff. There might be an exam question on there, but where I want to pick up with our focused review today is with some of those structural features of prokaryotes. So with these, like cytoplasmic membrane, for example, in any of these structures, you kind of want to have an idea of what they're composed of. So the composition here, you've got phospholipid bilayer, but also very importantly, you have some of those proteins. So the composition is important when we talk about the structures, but also what does it exam and not have a single idea what a cytoplasmic membrane on a bacterial cell would be doing to that organism. These six pretty straightforward. With the cell wall composition, we wanted to kind of leave block one with a pretty firm understanding of the difference between gram negatives and gram positives, because that's the way block three is set up. So some of the major characteristics between gram positives and gram negatives, structural characteristics, but also again, what does the structure actually do for that bacterial cell wall? And the fact that it has a lot of antigens on it could be used for antibodies to recognize and bind to. I have a couple slides. I threw in the endotoxin slide too because it's important. We'd had this on a check on learning question before, but it is important that you know which type of bacteria is associated with having, particularly like lipid A in it. When I say which type of bacteria, what am I asking about? Gram negatives, yeah. Gram positives do not have endotoxin. So lipid A is a component of this LPS, which stands for what? Yeah, lipopolysaccharide, which is in the outer membrane of gram-negative bacteria. It is super important. It's something we'll just keep talking about. And it's important because of this reason right here. If these bacteria start dying, especially if they're dying in large numbers and they release a lot of this endotoxin, you get some really serious health consequences for that patient. This slide is, just a little bit more detail. It actually explicitly says what happens when these gram-negative bacteria die very quickly before your immune cells have a chance to clean them up tidily so these toxic components aren't released into circulation. Capsule, super important. Is it on all bacteria? Some. What about gram-negatives versus gram-positives? Both. Yep, exactly. And what is it composed of? Sugars, yeah, for the most part, they are sugars. Is this antibodies are best at binding to in terms of macromolecules? No, what are antibodies best at binding to? Proteins. So if a bacteria is expressing a capsule that's covering its entire outer layer, it's a little bit harder. It doesn't mean it can't do it, but it is harder. This capsule is also kind of slippery. So when a big phagocyte, like a macrophage or something else, encounters an encapsulated bacteria, it's much harder to phagocytize it. Also, kind of intuitively, all those surface antigens that we talked about in the bacterial cell wall, if they're coated with capsule, it makes it harder for those antibodies and other recognition molecules to receive those antigens and respond. And also mediates adherence. So gram-negatives, gram-positives, and then this other class of atypical bacteria. It's a little nebulous, but we will talk about some important clinically relevant members that are considered atypical bacteria. Yes, sir.
On just in general throughout all these slides,
Mm-hmm.
should we be really locking in on the exact measurements of a lot of these microscopic things? Specific
Yeah, because the question could easily be, which of the following would be blah, 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 defining characteristic of an atypical bacterial cell. And if you thought it were in the ballpark, like an order of magnitude bigger, that could cost you that question. So yeah, in general, if it's on the slide, it's usually something you want to, because this is a defining characteristic of atypical bacteria. They tend to be smaller, so that's why it's in there, smaller than normal bacteria. Okay, so characteristics here, you want to know that they're very small. You want to know that something about their cell wall is different. It could be very, very different. There was a slide in that deck that talked about the differences between prokaryotes and eukaryotes, and we find prokaryotes, like bacterial cells, as having a cell wall. But there are some exceptions to that, and for example, mycoplasma is one of those exceptions. It's still bacteria, it still has the same genomic characteristics that make it a bacteria. It just happens to be really funky in that it only has basically a cytoplasmic membrane as a barrier with no wall. So that's one exception. It's a weird one. We will talk about it. There are some important pathogens in that genus, Mycoplasma. We'll talk about that in block 3. And then things like Mycobacteria, which is a different genus, completely different cell wall. We'll talk about that in much more detail later too, but that's why we call it acid-fast bacteria. Because they don't stain well because they have that waxy, thick cell wall. A lot of these do happen to be intracellular pathogens. Mycoplasma is another exception. Oddly enough, it's an extracellular, but most of them are intracellular. And most of them have unique metabolic criteria because of the fact that they're intracellular and they have access to things that extracellular bacteria oftentimes do not. So, just some general characteristics. Moving on to fungi, very straightforward stuff here, nothing too complicated. You gotta know a couple things about fungi. Eukaryotes and prokaryotes. Eukaryotes, exactly. So they're nucleated. If we were to break them down to these two basic forms, so all you have to be able to do is kind of understand the differences here. They're definitely different. With multicellular molds, for example, if you look at the structure of these molds, like here's a cartoon of some of the structures of these molds. These are specialized cells in a multicellular organism. So we didn't drill down to a lot of detail yet, but for example, we know that on a multicellular mold, these circles coming off these hyphae are spores, reproductive spores, right? So that's a specialized function. And the hyphae themselves have their own specialized function. They could be like these vegetative hyphae that are responsible for going out into the environment and obtaining nutrients, or they could be the reproductive hyphae that shoot up from the vegetative hyphae and actually support those spore-bearing structures at the top of them. So just some stuff to kind of pay attention to, basic stuff. Yes, sizes do matter. You know, the differences between, I didn't put the yeast slide on here, but generally yeasts, if you're trying to cultivate them in the lab, do they grow, does it take longer or shorter for them to grow relative to molds?
Shorter.
Yeah, they are quicker, they grow faster, generally speaking. Moving on to viruses, this slide had it's a very broad summary of their characteristics, but stuff you gotta know, basic viral characteristics. Important, you know, only have one type of nucleic acid. That means that in their genome, in their little virus structure, their virion, they're only DNA or RNA, they're not both. Size range, between 2 to 300 nanometers. I'm sorry, 20 to 300 nanometers. Genome, again, it's either DNA or RNA, pretty straightforward stuff. The exterior of these is important for classification reasons, knowing whether it's a enveloped virus or a non-enveloped virus. Another word for a non-enveloped virus is capsid or naked viruses, yeah. And regardless whether it's a naked capsid virus or an enveloped virus, there's always going to be some sort of viral protein covering the genome, keeping it protected. So that's called a nucleic acid. There's another, there it is, nucleic acid. That's specifically designed to protect the viral genome itself. In terms of going back to the outside of these, you see little like hash marks on the exterior of these virus particles. What's the significance or function of those? They're viral receptors so that they can bind to the exact type of host cell they need. Because if they get into the wrong cell type, it may not have all the requirements that that particular virus has. So they have specific viral receptors. That's what this cartoon is showing, usually glycoproteins. If we're talking about SARS-CoV-2, that coronavirus, we have a specific name for that viral receptor. It is called what? Spike protein on the virus. And then the host cell receptor that it uses is what? ACE2. Exactly, yep. So that means it's not, again, it's not just entering any old cell. It's entering cells that are expressing its cognate specific host cell receptor, ACE2. In terms of the major steps in viral replication, this is pretty straightforward. I have an analogy for it if you guys want one of my terrible analogies again. Yeah. Okay. The important, if you get a question about the order or of these events, it's important to kind of just think clearly through what's happening. There's also a diagram in your slide deck that shows from entry to egress, what's happening during infection of the cell. But it's kind of like if I want to go to my neighbor's house uninvited and use their kitchen and cook using their food and their cookware and their utensils and like their house, the whole 9 yards, and then I want to kill them afterwards. That's my analogy. Okay, so you walk up to your neighbor's door, that's your like specific, this is, Bob's house, type of recognition, this is the right place. I enter through, I don't know, kick down the door, whatever I'm gonna do, walk into the kitchen, then I gotta uncoat, so I'm gonna take out all the things I need. Not on this slide would be something like to suppress my neighbors from doing something, which viruses do oftentimes do, they suppress their host cell from doing something. But you gotta encode, so mise en place, get everything ready to cook, then you gotta start actually cooking, right? It's synthesis, the whole like complicated part of what you're doing. You guys have a pretty good handle on genome replication and what translation is and transcription, right? So all that stuff has to happen. And then afterwards, you have to like do all of the, put everything together, plate it, serve it to yourself, ostensibly. Don't let the host have anything, because that's not what viruses do. That's not the point. And then, before you get out of the house, if you're a naked virus, like I said, you probably want to kill the host. And if you're an envelope virus, you might just stay there until they run out of food and they starve to death. Okay. So. Yeah. That's my analogy for you guys. You're welcome. With the viral replication cycle, we talked about some important nuances in terms of what happens early on in an infection, from the time that you get exposed to a new virus to the time that you get like a significant viremia, large amounts of virus that are replicated successfully in tissues. So there's this big long lag period, can last between 1 to 20 hours in the eclipse phase. But during that eclipse phase, that's when all this stuff is happening. But not a lot of virus is getting produced yet, right? The food isn't ready yet. So 1 to 20 hours, you don't have a lot of progeny virus being released from those targeted host cells at that point. And after you do, then it gets kind of crazy. It gets crazy, release of virus particles, until or unless your immune system can kind of get a handle on things at that point. Exponential growth. Am I going too fast for you guys? It's just recap anyway, so. Okay. Prions, you gotta know what the nature of prions are. What are they? Prokaryotes, eukaryotes, viruses? Yeah, they're just proteins, right? They're your own proteins that are abnormally folded. In terms of transmission, there was another slide that talked about 3 different types of ways of acquiring prionopathies or prion-related diseases. Sporadic was one, acquired was another, and genetic. Yeah, exactly, inherited.
So with the prion is an unfolded protein. It's not like, because PRP is a normal prion?
It's a prion protein in the brain.
Correct. Okay,
Yeah.
so prion can be a folded protein before it actually goes through the unfolding process. I don't want to confuse the
No, it's a good point. So yeah, we have this PRP protein, and I think the gene that we all have is on one of these other slides. It's like PRNP R or something like that. So we have that. Something happens, one of those 3 transmission methods. Let's say you acquire it through eating beef. You get like the mad cow disease, the spongiform encephalopathy. Your normal prion proteins are fine until they come in contact with the PRP-SC, the scrapie form, the misfolded form, and then those misfolded proteins induce misfolding in normally folded proteins. Okay. That process is called conversion. It's unlike any other infectious process that we have talked about or will talk about. It's its own little, because these are proteins. So you're touching something abnormal is touching something normal and inducing it to become abnormal itself. And then in terms of defining prionopathies, these are just the diseases that prion diseases cause, that prions cause. They're defined as neurodegenerative disorders. Symptoms are kind of consistent with neurodegeneration. and they don't trigger an immune response. Those are all kind of important facets. There's not a lot of emphasis that you need to put on this slide. There it is, the PRMP gene. So if you inherit, like through that inherited mechanism, if you inherit mutated copy, you can get it that way. But in terms of pathogenesis, this is probably the most important part to focus on. There's not much to it, it's just basically saying what I just said. Once you have that misfolding, you have additional conversion, and then you start getting actual damage to brain structures. Okay, protozoan pathogens. With protozoans, all of these guys are going to be unicellular. They are classified by their mode of locomotion, how they move around. Pretty straightforward stuff. Make sure you got a good grasp on that. There's also a slide when we talked about protozoans about like cysts and trophs. Those are stages, they're not really developing, those are forms that are suitable for different types of environmental conditions. If you're in a rough environmental condition, would you be in a cyst or a trophozoite? Cyst, exactly. And if you're in a good, comfy place and you want to start replicating and eating, trophozoite, yeah. And then there's another slide on sexual reproduction, so I'll find it. With our worms, we talked about 3 different classifications of worms. This one is our trimatodes, or our flukes. What were the other 2? Cestodes and nematodes. Yep, exactly. These are also very straightforward. So imagine getting a question, like a simple question, where you're asked to kind of define what the characteristics are and make sure that you can remember what these broad differences are between those 3 different classifications of forms. Very straightforward stuff. Okay. Any questions on the first deck, on the intro to microbiology stuff? Okay.
So in the cartoon that was about viral replication, so it shows a capsid RNA going into the cell, going into the nucleus, right?
Yep.
And then it was an envelope DNA. Now obviously RNA doesn't need to go to the nucleus at any point, but an RNA can be enveloped or capsid, right? Like that can be interchanged, or is it always gonna be an RNA is a naked and a DNA is going on?
It's a fantastic question, and the answer is they are separate, and that's why I hate that slide, because it is sort of convoluted. You can have an enveloped DNA virus or a non-enveloped DNA virus, and you can have an enveloped RNA virus and an enveloped or non-enveloped RNA virus. It does not matter. That cartoon is actually trying to, it should just say, I wish it just said DNA versus RNA, because that's what it's showing. It's showing that DNA viruses need to go into the nucleus because they're DNA, so they'll use the host cell DNA polymerases to replicate their genome. With those RNA viruses, it's a different ballgame. They have to bring their own polymerase, because nothing in a eukaryotic cell can accomplish RNA replication. We don't replicate our RNA, so they have to bring their own, and ergo, they don't need to go to the nucleus, they just go to the cytoplasm. All this happens in the cytoplasm. We'll talk more about that in block 4 for LG. Yeah. Okay, so innate and adaptive immunity. There's nothing really specific pulled from the slide, just kind of like a nice one, because when we started talking about immunology, this was one of the early slides, and it might have confused you, but now that we've been through the entire immunology deck, hopefully this is sort of pulled together a little bit better. It makes more intuitive sense. The main point of the slide is just kind of showing you the main players of the innate immune response, and then sort of like a rough timeline, even though we talked about this in more detail, of when that slowly transitions and hands off to the adaptive immune response. And then again, what the major players are in the adaptive immune response. You've got B cells, then you have plasma cells that start secreting antibodies. We've got T cells that are programmed for specific antigen recognition. They get activated, they proliferate, and they begin to become effector T cells. Skipping ahead a little bit, when we say effector T cells, we talked about 2 really, really big, broad classes of T cells that have different effector functions. CD8 and CD4, exactly. And which one were the helper T cells? CD4, and then the cytotoxic ones were CD8. Very good. Whereas B cells, I mean, they're moving into that deck a little bit more, early on, there was an intro slide for the innate response, there's another intro slide for the adaptive immune response. Kind of set these up to mirror each other in terms of just giving you a broad overview. So it's nice to kind of hit these and study, you know, what the major players are in both of them, what they're primarily doing, and then how they relate to each other, at least the innate response, how it's like transitioning and handing off to the adaptive immune response. And then the adaptive response itself, like the primary initial exposure to an antigen and how that's different than the secondary exposure after you've seen him. So just intro material, but good stuff to study. This too is a nice slide study. I like seeing stuff like that. It helps you kind of get your head around lineages for these lymphocytes, or leukocytes I should say. You certainly do not need to memorize this stuff. It's just sort of there because you're going to see it throughout IPAP in different courses. What the normal range is of these different leukocytes. But yes, this is primarily, these are the cells that we're primarily focused on. The cells of the innate immune system, which includes natural killer cells, and then the adaptive immune system, B cells and T cells. We talked about physiologic and chemical barriers of the innate immune system. Straightforward stuff. So just be kind of prepared to answer questions about those barriers. Here too, what defenses do, what lysosomes do. And then talking about these innate cells separately, sort of baseball card style. Where are they usually found? What is their function? How long do they live? Stuff you want to pay attention to that can be helpful. You know, what they're best at doing. A lot of these guys are multifunctional. It says right here for macrophages, we talked about the versatility of macrophages, the fact that they do a lot of things, and, you know, produce cytokines, they do tissue modeling. They don't just clear pathogens, they clear cellular debris as well. They do a lot of things, but their best thing is eating phagocytosis. Neutrophils, likewise, these guys, you want to know where they are normally found, where you usually get, where most of your neutrophils in blood. Yeah, they're circulating. And there's a huge component, a huge percentage of all your leukocytes are neutrophils. So since they're just circulating in blood, they stay there until they're recruited or attracted by some sort of chemoattractant. And then they move through chemotaxis towards the thread en masse to solve those problems. How do they solve those problems? What their capabilities are? They are also big eaters, big phagocytes. They're super dangerous. In addition to phagocytosis, they can degranulate. They can do this mitosis function that we talked about. It can be really damaging to host tissue as well that's around that environment. So important to remember that. But importance in immunity, always make sure you kind of pay attention to that too. That these things are recruited to the site of injury or inflammation. Yes, sir.
So just to clarify, so mitosis is basically anthropomorphized neutrophil. It goes in, it does a common causing attack into the area, and it just kind of splatters, and then pathogens will get that granules and protein.
Yeah. Yes. So the kamikaze nature, like the bad ending for the neutrophil, is actually common among all these different methods. It's a one-trick pony. When it encounters something and it kills it, it's like a wasp stinging a person. It dies afterwards. It's going to die regardless. Mitosis is just a little bit more of a dramatic way to go out, because during the mitosis, you're right. Like when its genome is getting decondensed, and when the histones are being flung out and all those membranes are dissolving, that is mostly, it's a physical way of killing those things by trapping them so they cannot move. And there's also some toxic components of the interior of that neutrophil that will also contribute to the death, particularly of bacteria. So it's very, very effective at killing bacteria, particularly. Yeah. Yes, sir.
So each neutrophil is just a one-time use cell?
Yep.
Regardless of how.
Pretty much. So even if they don't ever in their little legs encounter any pathogens, they are not going to live much longer than 3 days. So anywhere between less than a day up to 3 days, they're very, very short lived. That's why you make so many of these in your bone marrow. You're pumping out huge numbers every single day. Yeah, that's why you're doing it, because they're designed to be dangerous little killers that should be used sparingly, but in good abundance. And then here, granules containing antimicrobial substances, like that's part of the nastiness that they unleash when they're killing. So this was my first practice question. It is, like I said, they're a little bit harder than what I would give you on the exam. This one is like a clinically oriented one. But as you're reading this, the first sentence, we haven't talked about anything really medical or clinical yet. And some of you guys I know have no backgrounds in clinical or medical stuff. So I wouldn't be asking you something like this on an exam per se unless we talked about it a little bit in lecture. But let's say you did get a question like this. You do not need to freak out about this stuff. Most of you have heard of chemotherapy. Anybody know what this term means, neutropenia? Low levels of neutrophils. Yeah. So, but we don't know that. But let's see if we just had the second part of this, like the actual question part, if we could figure this out based on what we already know about neutrophils. Which of the following is the most important mechanism by which neutrophils contribute to the resolution of a bacterial infection? What do you guys think? What's hearing a lot of delta, lymphophagocytosis, and that is correct. But say you didn't know that, could you eliminate some of these just by being ridiculous? Yeah, they're not immune suppressing anything. Yeah, no, they're out. Yeah, definitely eliminate E, right? That's ridiculous. What else is ridiculous? Antibody production, yeah. What produces antibodies? B cells, yeah, plasma cells. Antigen presentation. If you're only living for 10 hours and then you die after contact or exposure, would you be a good APC? No. No, they're not. They do have some MHC, but they're really, really crappy APCs. They're not considered to be professional APCs. So then it down to a 50-50 complement activation versus phagocytosis. Complement activation is not a primary function of neutrophils at all. They phagocytize, they destroy. So, okay, so that's not so bad, right? I guess the point more than just giving you a practice question is to kind of get you to be more confident when you encounter a question that you think immediately is going to be a harder question. It usually isn't. You just have to think about it and kind of break it down to the easiest possible way. Eliminate things that are ridiculous. Focus on things that are possible. And then here too, I got another one for you. Patient who develops a localized staph aureus skin infection, which of following immune cells would be the first to be recruited to the site of infection in large numbers. What do you guys think? E, the neutrophils again, yeah. Yeah, so what did you think?
Oh, they're in the blood, right? They're in the
Yeah,
blood. So
exactly.
their macrophages are already there in the tissue.
Yes. So when I said, like, when you're studying this, let's go back to macrophages. Where do they live? They're already there. Yeah, they're not getting recruited. I mean, they might, like, locally move a couple cells over or whatever, but things that get recruited from circulation are these guys, and they get recruited in large numbers. Okay. So, ridiculous, 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 ridiculous. Good answer.
Just know it.
Always, yes. Always right. Done. Yeah. Okay, so et cetera, you guys get the point. T cells, same thing. They're very best at doing what? Like the number one ABCs. Yes. Doesn't mean other things can't present antigens as well, but these guys are super good at it. Chlorophyll is function. They've got all these funky tendrils sticking out all over the place. So they're very good at that. And then what about where do they live most of the time? Where do dendritic cells like to concentrate? Yeah, definitely in the intestines, but more broadly. Barrier tissues, yeah, so all those mucosal parts of your body is gonna be, packed full of dendritic cells. Very important for T cell activation. As these are. Okey-doke, the ones that are jammed on one slide, won't do again. These are the granulytic cells, that are oftentimes involved in allergic responses, inflammation, particularly with eosinophils, importantly, parasite defense, very important role there. Things to pay attention to, they're all relatively rare cells, mast cells in particular are like, just extremely rare to see. Bone marrow, always when you're studying this, study commonalities and differences. So bone marrow, mature and connective tissue, so that's a difference between mast cells, and one of the differences between mast cells, basophils, and eosinophils. Mechanism of killing, what else is important? But yeah, mostly roles, and these do have very similar roles. They're all gonna be involved to some extent in allergic reactions and inflammation. Another question for you guys, I would not ask you this, not an exam one, but let's just see what you think. You've got somebody with a history of asthma, they have a bronchospasm after exposure to some allergies. Which of the following cell types plays a central role in the immediate hypersensitivity reaction? Oh, we got some. Yeah, I'm hearing people say mast cells. Why mast cells? Yeah, because everything else is ridiculous. Natural killer cells. What cells of the adaptive immune response are natural killer cells sort of like in terms of their killing mechanism? They're a lot like cytotoxic T cells in their killing mechanism, CD8 T cells. Something very similar. The main difference is their way of targeting or zeroing in on those infected cells, which are usually oftentimes infected with viruses. They're very good at killing viruses. Initially, before T cells have a chance to become activated, these guys would be the ones that would be clearing those infected cells. And they do that by virtue of recognizing something that's aberrant about those cells that are infected. Okay, another question for you. So, patient with a viral infection, the virus is doing its usual manipulation of the host cell, and it's causing that host cell to downregulate expression of MHC class I on the surface. Which of the following immune cells would be most effective at killing those infected cells? Natural killer cells, yeah. Why not CD8 T cells? Why not the cytotoxic T cells? That's what they're good at. Yeah. So what does a TCR bind to? MHC1. So if it's downregulated, if it's not expressed, you gotta really rely on natural killer cells. If a person has some sort of immunologic or some genetic defect in natural killer cell function, they are gonna be screwed for that innate rapid response to viral infections. Did you have a question? Okay. Yeah, natural killer cells. Alright. Soluble factors in the innate immune system. We talked about complement a little bit, we talked about cytokines, antiprotopeptides, defenses, the things that are very good at inserting indiscriminately into bacterial cell walls and causing them to lose structural integrity. But let's talk about cytokines briefly. Overall, the broad purpose of cytokines, what are they doing for the immune system? Facilitating communication, they're signaling, activate, deactivate, suppress, all that stuff. They're sending signals based on what is happening to the cells that are secreting them. So all these different classes of cytokines are doing different things. Talk about the complement system. We had complement, or these are soluble proteins that have three different major effector functions. One is lysis. Here in this part of the cartoon, lysis is being shown where complement is binding to bacterial cells. It's forming what we call a membrane attack complex, and that's causing those bacterial cells to die. There are also specific components of the complement complement system can freely float off either locally to attract and recruit other immune cells like mast cells, or they can go into the bloodstream and start recruiting things like neutrophils to come into that area, diaphoresis of these neutrophils into that area through chemotaxis. And then they also have a role in opsonization, which is what? Yeah. Sugar coating, yes, making things more tasty for phagocytes. Yeah. So coating these, in this picture, we're seeing complement receptors on some sort of cell that needs to go. So it's coating those complement receptors, and then antibodies are piling on for a synergistic effect to make it easier for phagocytes like macrophages to come and clear them. Or in this other mechanism, we've got complement bound to a bacterial cell, which makes it easier for direct phagocytosis. All right, let's have a crack at this one. So here we've got bacterial infection that we haven't talked about. We don't really know anything about. I think I actually did talk about Neisseria a little bit, but regardless, we haven't covered it formally. This patient has recurrent Neisseria infections, and they have some sort of genetic deficiency where the late complement components C5 through C9 are not working. We didn't talk about those, but let's just have a crack at this. If they have this genetic deficiency in their complement system, this would impact which of the following functions do you think? Yeah, cell lysis. Yeah.
So it's the end of the complement is what typically like the later ones fill the back.
Yeah,
Right.
exactly.
That's kinda how
Yeah, exactly. You said those later complement components are the ones that are responsible for forming the MAT complex. And that's what this is. Cell lysis, that's the formation of MAT complex and bacterial surface. What did it say? Yeah. So, yeah. If opsonization were an option on there, that would be a super bad question. So. Okay, cytokines, make sure you kind of understand this slide. We've talked about this a little bit, what they basically do, but they're signaling molecules, and that's what this cartoon is showing. You get some sort of information coming into one cell, like, hey, I'm infected by some virus. You've got to let your neighbors know. You secrete cytokines, and you tip them off that something's going on, so they can start preparing, and they can start signaling. They have some sort of effector function that's appropriate. And this target cell could not just be a neighbor cell, it could be an important immune cell as well. It could be activating T cells, it could be activating B cells, it could be activating macrophages. All of those things take this sensory input from cytokines. The antigen presentation, yes?
Are the cytokines being released mostly by the innate cells, or is that also like B cells and T cells?
Great question. And adaptive immune cells do as well. They do a lot of complicated, sophisticated signaling, talking to each other, especially T helper cells. Yeah, pretty much all cells. I mean, I'm focusing on immune cells because they need to convey messages, but even epithelial cells have some cytokines that they can release as well to communicate what's going on because they're epithelial cells, they're getting infected too. They need to, it's that bystander, if you see something, say something, everyone needs to be able to communicate that there's a problem or an initial problem. So, some cells just do it more robustly than others, but all cells have that basic, endothelial cells, fibroblasts. Antigen presentation, this slide had a little bit more detail, but it's showing us the difference in the mechanisms of presenting antigens to two different types of effector T cells, our CD4 cells and our CD8 cells. With our CD8 T cell activation, here we have what kind of MHC that's being recognized? Type 1, and what kinds of cells would be capable of expressing MHC1? All nucleated cells, yes. So any cell that gets infected, for example, is going to be processing pieces of that pathogen, presenting it on MHC class 1, and then the MHC class 1 will interact with what molecule directly on T cell? TCR, the T cell receptor, exactly. So that's what that orange little thing is right there. only interaction that needs to occur between the APC and the neural, or the effector cell. Yeah, so what is that? CD8, yeah. So look at this cartoon, CD8, that is coming up and it's touching MHC class 1. If that were not expressed on that cell for whatever reason, do you think it'd be able to facilitate that interaction? If it didn't have MHC1? No. And that's why when it's downregulated, for example, natural killer cells are the way to go, because they wouldn't have any MHC class 1 necessarily to be downregulated. But these guys, it's present, so they're recognizing it, and they're able to do their effective function, which is essentially to kill that infected cell. If you contrast that with an antigen-presenting cell, what molecule is it presenting? So this MHC class 2, that's right, but it's not infected, at least not necessarily. It is a special type of cell. What type of cell would an APC be? Things like macrophages would be good, dendritic cells would be really good, APCs, like professional antigen-presenting cells of the innate immune system. They're the ones that are going out there deliberately phagocytizing entire bacterial cells and things like that, processing it, presenting it, and then sharing it so that those CD4 cells that express TCRs and CD4 can recognize that and behave accordingly. Okay. Yes, sir.
You said that it initially makes contact, like the MHC1 receptor makes contact with the TCR?
Yeah, MHC on the cell that is infected or an APC. And then on the T cell, it's the TCR, it's a T cell receptor.
T cell.
That's the interaction. And there's a couple good slides in your deck that kind of show that primary interaction that's going on. But that is when that virally infected target cell or the APC is actually presenting the specific antigen to that T cell in both cases. You see that squiggly line here? Here it's like the chopped up piece of virus. Here it's the chopped up like bacterial peptide that was processed and presented. But in either case, it is physically getting like loaded onto that MHC and presented to the T cell.
Is this where the super antigen takes away the CD8, CD4?
Yeah, so it's related to that. There's it's not, I highly recommend you review this. I don't know if I put it in the review deck, but what the super antigen would do would be, instead of binding in that specific cleft on the MHC molecule where it's supposed to be, that specific antigen recognition cleft, it would bind on the exterior, which is non-specific, and it would cause a bridge between, like a semi-permanent bridge between the presenting cell and the T cell, which would cause in turn uncontrolled stimulation of the T cell. Okay, I've got 1350 right now, 1352. We've been going at it for a while. Why don't we take a 10-minute break and pick back up at 1402 with this slide. Yeah, it's
Get home telling the fucking trip.
Oh, yeah. Yeah.
Yeah, but yeah, they had all like, in terms of it's okay guys. I've got 14:03. Alright, how are we doing so far? Give me like the brutal honest, is it okay?
No.
What time are you hearing this?
If you just give off more cytokines, that's gonna be on the test.
Okay, yeah, I'm having trouble reading this room.
I would be spending many hours.
Oh my.
That's good, huh?
The flashing red lights going
I
off.
didn't hear that, but I probably didn't want to hear that.
It was really funny. Whoever said it was really funny.
So, alright. Well, just know that, like, if this is all just, well, if it's all like this, and it's not a good prognosis, but, I have office hour, I'll be here tomorrow, and I will be here on Thursday, and, you know, if it helps, if you wanna just go over terms, sometimes I think the best thing to do is just, in your own words, try to describe something on a slide, and make sure that you kind of, it makes sense to you. And I, you know, if you're doing that in your own study groups, that's tremendously helpful. Or just going through slides and generating questions and making sure that you can answer them, okay? So like I said, just let me know if you wanna talk, Wednesday or Thursday this week. Okay, so moving forward, we're on whatever slide this is in depth 2, where we're talking about specific features of the adaptive immune system. We talked about those 3 features. This slide only has one, the most important one. When you're talking about B cells and T cells, they have those antigen-specific receptors, BCRs and TCRs, respectively. Which in both cases, upon maturation, is pre-committed, it's fully programmed to recognize one antigen, and one antigen only. Other characteristics, just defining characteristics of these B cells and T cells is that they have the capability of, after the fight is over, after exposure, after encounter, of long-lived response, longer-lived existence, and the ability to proliferate and expand as needed if they get re-exposed. So the same T cell that saw an infection 15 years ago sees the same type of antigen again. It needs to be able to receive those signals, a lot of times from cytokines, signals that tell it to proliferate again, and basically it's like going back into battle again as a veteran this time. The basis of memory. And then immune tolerance is another cardinal characteristic of these cells of the adaptive immune system. They need to be able to, upon maturation, know what is and is not self. Otherwise you get autoimmunity. Organs of the immune system, we talked about 2 primary organs. We talked about bone marrow, and we talked about the thymus. The thymus, easy to remember with T cells, T, thymus, right? B, bones, B cell, bone marrow. Even though that's not why I was named B cell, but yeah, important for both B cells and T cells to mature. But we spent more time talking about secondary organs of the immune system, talking about the lymph nodes primarily, and these mucosa-associated lymphoid tissues that are throughout the body. They're just packed with immune cells. And the spleen as well, just like the lymph node for the blood. hated this slide, got it. But it is super important to have a familiarity with, hopefully with CD4 and CD8 surface markers on T cells, you sort of have that justification in your head after we've talked about it again today, like what it's doing. These are defining features and markers for those cells. CD3 is a less specific marker. It's present on all mature T cells. CD8, which is important to receive co-signals on APCs. That there are some surface markers that function for adhesion. And then on B cells, those two have different markers as well. They're APCs, so they have MHC class 2, for example. Would you see MHC class 2 on a T cell? Nope, you would not. They're not APCs. And then this slide is nice because it kind of shows you all those different surface markers and kind of specific roles for them in adhesion or the primary receiving of antigen-specific information, co-stimulation signal that you get between CD28 and B7, and it's cytokine signaling. These T cells will also have receptors for cytokines to receive that information as well, and to send that information. Okay, practice question for you guys is somebody's researching a new vaccine candidate. Which of the following cell surface molecules in dendritic cells is most important for providing co-stimulatory signals to T cells, leading to their activation? One right answer.
B7.
B7, yeah. Yep, it's B7. MHC1 and 2, definitely not what they're going for. Those are not co-stimulatory signals. CD3 is what? We just said what CD3 was? Mature marker, yeah, it's responsible for signaling some other roles, but it's not a co-stimulatory signal. So that leaves between CD28 and B7. If we're talking about the APC, if we're talking about the dendritic cell, then we're talking about B7.
Can you go back to slide? I'm sorry. Can you go back to me?
Absolutely.
Or another one, I'm sorry.
Yeah, so B7 will be on the APC, like dendritic cells, the mother of all the APCs, right? The best APCs out there. And it's binding to CD28, which is a surface marker for T cells. And then here you can see that's exactly what this third panel is showing here, co-stimulation. You got CD28 on the T cell, and it's receiving that co-stimulatory signal from your dendritic cell, your APC, whatever. Yeah, by expression of B7. If B7 isn't on that APC, if that dendritic cell doesn't have it, probably not going to get stimulated to have a response. You would have T cell anergy at that point. So, the right answer is B7. Okay, T cell subsets. Kind of framing, there's a lot of text on this slide, so I'm going to break it down and make it relatively simple. Think of helper T cells and all those subsets as being communicators and coordinators and orchestrators. That's all these subsets are doing in different ways. Look at those action verbs. Th1, activating. Th2, activating. Th17, recruiting. Tregs, suppressing. Those are influencing roles, and they're doing it primarily through cytokines, through expression of different cytokines, and signaling. That's what helper T cells do. They have an interaction with CD8 cytotoxic T cells, but they will also help to activate B cells as well by those interactions. So that's the way I want you to think of all these subsets of T helper cells. They're communicating and activating and suppressing and recruiting, those types of functions, helping out. With cytotoxic T cells, we've talked about these a bit. Again, you want to think of them, at least in terms of their killing, cytotoxic T cells, much in the way you think of natural killer cells. They have the same mechanism of killing, just different ways of getting activated or stimulated. Okay. So make sure you understand that and study that. Alright, another practice question for you. Got a patient who's HIV positive, so their CD4 count is dipped below the clinical definition of AIDS, which is below 200 T cells, CD4 T cells per microliter. The patient is at increased risk for opportunistic infections due to deficiency in which of the following immune functions, do you think? Basically, that question is asking you what best describes the function of CD4 T cells, the helper T cells. Yep, exactly, cytokine production. Of this list, rule out the ridiculous ones, they definitely don't produce antibodies, right?
Definitely not.
100% not.
Yeah. Complement activation, we've not talked about that as a primary defining role for CD4 T cells. They're not phagocytes, right? No, get rid of that. Direct killing of infected cells, what T cell does that? CD8 cells, get rid of it, it's ridiculous.
So the CD4, in it releasing its cytokines, is it transitioning to these other T helper versions of itself through autocrine, like, or is it only is it getting that development before it's releasing and it's, I guess,
Yeah.
is it naive when it's
Naive means that you have not yet encountered or
Right.
inactivated.
Okay.
You have not encountered your antigen. You haven't stumbled across it. When you encounter that antigen through an ATC interaction, then you become activated and mature. And then you can differentiate into some subset in there.
but that differentiation is happening from itself, like it's
No.
it's from its own cytokine.
Not
cytokines?
Not entirely, but it can be. So when you're self-signaling, there's a word for that.
Yes, autocrine.
Autocrine, right. So it will receive some autocrine cytokines like IL-2, for example. But it will also probably or potentially receive other types of signaling, cytokine signaling, such as local, which is called paracrine, or at a distance, which is called endocrine signaling. So it's not necessarily all autocrine signaling and receiving of your own secreted cytokines. It could be messages from other cells as well. Does that make sense? Okay. John, you have a question?
Yeah, couldn't you make an argument for D though? Because since Th1 activates macrophages, then phagocytosis would be down to it.
Can you make an argument for what?
D as the answer.
Phagocytosis?
Because if Th1 activates macrophages and now you're not activating it, then it's going to lower phagocytosis.
And that's a good argument. So a couple things with that. It would be indirect. It's not a primary mechanism. It's certainly not the best option if it were between phagocytosis and cytokine secretion, no contest. And thirdly, macrophages, they could be activated by other ways as well. Yeah, so it's a good thought, it's an excellent thought, but definitely this is the better answer.
It's a ridiculous thought.
It's ridiculous.
Well, it's not ridiculous. It's a great thought.
It's ridiculous.
And certainly they themselves are not phagocytes. So, yeah. Okay. T cells, the humoral response. In order for, so these are antibody functions. We talked about a bunch of different effector functions for antibodies, what they do, how they're able to clear threats, microbial threats. As we talk through this, for all these different functions, these functional categories of what they're doing, what do they need to be, and we'll go through structure really quick too, what do they need to be able to do in order to have that effective function? You have to have binding, right? So that region up here, which is called which region? The Fab region, it has to have an exposed epitope. So that doesn't bode well for an intracellular pathogen, does it? It's not, an antibody isn't gonna go like penetrating through an epithelial cell that's infected with a virus and go binding, digging around in the cytoplasm to find it. No, it has to be exposed. So because of that reason, the humoral response tends to be better at clearing extracellular pathogens a lot of the time, and things that it can see, quote unquote. By see, I just mean the Fab region can actually reach out and touch and bind. And real quick, in terms of how B cells are developed and activated and matured, these two have a pre-programmed BCR once they leave the bone marrow. that BCR is responsible for exquisite antigen binding and internalization through receptor-mediated endocytosis. There's a nice figure that shows that. And then it does need T cell help. So you're not gonna get appropriate B cell activation unless you have that handshake from T cells, helper T cells, yeah.
This may be a silly question, but is the BCR the antibody before it's released?
No, the BCR is, it is an antibody. It's that Y-shaped membrane-bound molecule, but it's not going to lose its transmembrane domain itself and get solubilized, cleaved off and solubilized. There will be some sort of signal that tells that cell to start upregulating soluble forms of antibody that can be secreted out.
Okay.
Yeah, but the BCR itself is it's a membrane-bound antibody. Okay, on this slide, yes, sir.
So the B cell is most commonly like coming into contact like with antigen free out and about.
Oh, that's a great question. Okay, so the B cell itself, it is an APC. Remember, it has that APC function, but it's a little different than the other APCs that we talked about. We talked about macrophages and dendritic cells, right? Being really good at it because they can phagocytize it and engulf, they can do all these things. B cell meets its antigens differently. It usually gets handed them like a handoff from these APCs, so it's kind of indirect because it's not phagocytizing. So the dendritic cell that's out patrolling around some mucosal surface captures something, it presents it, it'll show it to a B cell. The B cell will receive it on its BCR, and then in turn do whatever it's going to do if it recognizes that antigen specifically. But usually it's through presentation through another APC. And then it in turn has its MHC, so it will do its thing, process it, and then present it on its own MHC to what kind of cell? Exactly, CD4 T cells, yep. Okay, B cell development and activation, just kind of read through this text a little bit. I will not go into a lot of detail, I just want to make sure that you guys kind of understand this text. And I find this cartoon to be extremely helpful in understanding what is going on with B cell activation. And where is most of this B cell activation occurring? Where are these B cells getting activated?
Bone marrow.
Not activated, they're maturing in the bone marrow, but then
Secondary.
to the secondary lymphoid organs. So they're going to the lymph nodes and they're going to the spleen. Okay. Once they're in the spleen, once they're in the lymph nodes, once they're in the vault, that is where all these different APCs are coming back with all these things to show them. Here, look what I found. Look what I found. Check this out. But it's happening in secondary lymphoid organs. And they need T cell help again.
I got a question.
Yes, sir.
So they go to the secondary lymphoid organs and they're presenting to what?
The B cells themselves, so once they leave the bone marrow, they have their BCR, right, the membrane-bound antibody. They're going to the secondary lymphoid organs and they're going to wait there. They're going to wait there for a long time. If they never get presented with some antigen-presenting cell that says, hey, look what I have to show you, that's fine, they won't be activated. But if they do come across an APC that's like, yo, I have this case of Streptococcus pneumonia, I want to show you, look what I found, you know.
And B cells can present that to other B cells, correct?
B cells can present that to T cells, to CD4 T cells. So they'll receive that antigen from some sort of like dendritic cell or APC, another APC. They'll process it themselves, and then they'll tell the T cells, CD4 helper T cells, they'll tell them by virtue of their MHC class 2 onto the TCR, they'll tell them what they found.
And that's for the memory T cell purposes, right? Or is that, what is the T cell?
No, so that interaction, so now the B cell is basically just, it's in APC mode, it's sharing its antigen with the T cell. That CD4 T cell is being activated. So it still has, that's all that's happening is being activated. It doesn't mean it's going to survive and become a memory T cell yet.
So once the B cell presents it to the T cell or the helper, right?
Yep.
CD4, is that when the T cell subsets, where they decide if they're gonna activate macrophages, activate eosinophils, etc.?
Yeah, and that's a good question. So all that T cell needs is basically it needs to have the right TCR, the right T cell receptor to receive that from the MHC2 on that B cell. If it does meet that and it meets those other interaction requirements that we talked about previously, co-stimulating signals, etc., then yeah, it will further differentiate according to what kind of antigen was presented into a Th1, Th2, Th17, Treg. Yeah. Okay. Antibody structure. This stuff I think most people have a pretty good handle on. But when we talk about the structure, you kind of just got to know a couple things. Fab region, that is the part that's like the Y part of the molecule, the part that bifurcates or forks out. The stem is the Fc region. It stands for constant, but it's not really constant. It does have a lot of variability as well. The Fab region at the very end has those variable regions that are super variable, as in different combinations of them are responsible for being able to bind specifically to basically like any antigen imaginable. Okay. Other features, you've got this big heavy chain that is 2 to 3 times as massive as the light chain. The light chain only extends along the arms, so to speak, on the outside, if you will. But the heavy chain will extend all the way from the tip of that variable region on the Fab all the way down to the feet, the stem of the Fc region. It has a hinge region in the middle right there. It is linked together with these disulfide bridges. And then in a monomeric form like this, it's capable of binding how many antigens? 2. One on each one of those arms. One antigen and another antigen. And it would be the same antigen, just like it's like a mirror, so same specificity as possible. Then we talked about classes of antibodies, 5 classes in humans, other animals that are in process. Don't worry about IgD so much. IgG is really important, and IgM is super important. Well, everything about IgG is super important. The first one that gets produced or secreted from an activated B cell or plasma cell is which one? IgM. And that has structural drawbacks and advantages. It's super big, but it also offers a lot of avidity because it has all these antigen binding sites that it has. So, yeah. A couple terms to know what an epitope is, how it's different than an antigen. What would you say, like if you had to describe the relationship between an epitope and an antigen, what would you say?
Epitope is lumped together.
Epitope is the very the small distinct part of the antigen that's recognized. And like either an antibody or like the receptor on the APC. Like it recognizes the epitope.
Yeah, on a T cell.
On a T cell.
TCRs would recognize a super specific epitope or a Fab on an antibody, one of those two. So, and this cartoon is excellent at showing that. You've got this big antigen, even though it's not that big, but it has 1, 2, 3 on this cartoon, 3 different epitopes, although in real life, any one of these could be an epitope. But those epitopes are the pieces of that antigen that are specifically recognized, oftentimes by antibodies, but it could also be a T cell receptor, TCR.
So would it be like the antigens like molecule that epitope, I guess you could specific sites and let it find those?
Yeah, that might be a good analogy. You don't like my lock and key analogy? No, I'm sure that's fine. But yeah, like total. Actually, that's a really good analogy. Kind of the difference between me just kind of looking at you and saying, yes, I've recognized you, I know who you are, versus me doing like a full DNA analysis and match and biometrics and confirmation of identity. But with the epitope, it's like that exquisite recognition with absolute certainty. Okie doke. Another super long, like I said, these are harder than I would ask on the exam because we haven't talked about ELISAs, but Lyme disease, many of you have heard of Lyme disease. It's a bacterial disease, we'll talk about it in block 3, but it's hard to diagnose. So patients oftentimes come in, you could take blood from that patient and order some antibody testing to see if they've raised antibodies to the bacteria that causes Lyme disease, Borrelia burgdorferi. So they'll use an assay called an ELISA, which we'll talk about next block. And they'll incubate that patient's serum that either does or does not have antibodies against Lyme disease in it, with antigens from this bacteria, Borrelia burgdorferi. If they have it, which part of the antibodies in their serum will directly bind to the bacterial antigens coded on the ELISA? The variable nature. So all this extra fluffy part, you could just reduce all of that down to saying, what's the epitope recognition part of an antibody? It's the Fab. So again, like when you see horrifying questions, just try to cut through to the part that is like the direct question. Okay, research team is developing a novel therapeutic antibody to neutralize the toxin produced by C. diff. To engineer an antibody with high affinity and specificity for the toxin, which region of the antibody molecule should the researchers focus on modifying? And there's the answer again. Yeah. So, same question. This is just a little like another way of asking that you know what part of an antibody recognizes the specific antigen. All right. We talked about class switching a little bit. This is when, over time, those B cells, once they become activated, and once they become plasma cells, they will initially produce what class of antibody? We already said, IgM. And then after a while, levels of IgM will taper off. Those are called titers. Titers of IgM will taper off. And you'll get signals that tell that plasma cell to undergo class switching, which means you keep the same Fab, but change the Fc region. Change it to a different class. It does a different thing. So we're no longer going to produce IgM, we're going to produce the same Fab, same site picture, but we'll just make this monomeric IgG because IgG has features that IgM doesn't. It's longer lived, it's more stable, it's smaller, it's capable of going into places that big pentamer can't go. So that's why we undergo class switching. But the main point is, again, you keep the antigen specificity. Okay. Which of the following antibody isotypes, if they would provide strongest evidence of a recent infection of the CNS by a pathogen? A recent infection. And why would it be them? Yeah. And that's actually oftentimes used to answer a clinical question. Does this patient have a recent exposure, or did they have an exposure to something 20 years ago? IgG titers will drop off over time, but if you were immunized against measles, you had your MMR as a kid, you're not going to have any IgM. That would be evidence of a recent infection. You would have IgG. And that's what people would look for. You look for IgM versus IgG. Okay, effective functions, make sure you're comfortable with neutralization, which is one of the key effective functions of antibodies. What they're doing when they're neutralizing pathogens and toxins. Sorry guys, too many practice questions. Okay, so say you vaccinated someone against tetanus, which is a disease caused by Clostridioides tetani, and it is mediated by tetanus toxin. What is a primary mechanism by which the generated antibodies protect against the toxin, would you say? pretty straightforward. It is the toxin is going to be neutralized by those antibodies that can bind specifically to it.
Remember the silly ones and why are they silly?
Okay. Yeah. Okay, so we're talking about antibodies, we're talking about the humoral response of B cells being able to produce antibodies. That's what this question is asking about specifically. And then the threat is a toxin. So B cells do not primarily activate complement to lyse to bacteria. That's not what they do. We should be immediately kind of zeroing in on antibody-related options. So we can eliminate A. With B, are we enhancing phagocytosis by the humoral response? Are we trying to really stimulate macrophages? Not really, no. Let's skip down to D. Stimulating the production of reactant oxygen species by neutrophils. Do B cells really have a role in that? Not really, no. Nope, that's like way down on the innate level. Neutrophils do fine on their own getting there. And then lastly, inducing apoptosis of Clostridioides tetani, the bacteria that causes tetanus. What does that sound like? What other cells would be responsible for inducing apoptosis? More like CD8 cytotoxic T cells or maybe natural killer cells?
Does that mean that like death ligands like it causes apoptosis?
Exactly. Either through one of through one of two different pathways, they ultimately, even if you're dumping your enzymes through a whole truck caused by programming, either way, that target cell, that infected cell, is going to die by apoptosis. But that's describing to a T the mechanism of killing for CD8 T cells and natural killer cells. So that's why I would kind of eliminate those four other options. Yeah.
Right, because we talked about one of the functions of B cells or antibodies, I guess, is oxidation.
Like, are we going to during a review? Or we did, yeah. And opsonization is on the slides. It might not have made its way to the review deck, but yeah, it's a function that you should definitely.
So why would enhancing phagocytosis
Yeah. Well, it is talking about the toxin itself for one thing. So if macrophage is coming along and trying to phagocytize all the toxin in the body, it probably wouldn't be like as good of a mechanism as antibody-mediated binding to that toxin. Yeah, for toxin,
So you're talking more like efficiency?
Yeah.
It would be inefficient to phagocytize everything versus just blocking it?
Yeah, especially for, consider the urgency and the timeline of this. Say you have a toxin-secreting bacterium, like Clostridioides tetani. It's secreting a lot of toxin, or Staph aureus can produce a lot of toxin, become really quickly dangerous. For immediate responses, or at least the ability to very quickly neutralize that toxin, antibody would be a good way to go for that. If you've had previous exposure, like a lot of our vaccines that we've talked about, Tdap, DTaP, those are built so you have antibodies to tetanus toxin already. So if you were stuck on a rust nail or whatever, rather than have to mount your own immune response, you already have antibodies against tetanus toxin. If that makes sense. And that's a really efficient way to help clear that threat.
So like neutralization is against the toxins that are secreted by the pathogen, and then phagocytosis is attacking the pathogen.
Neutralization isn't just for toxins. So neutralization, there's a couple of sub-mechanisms that we talked about as well. Toxin neutralization is one of those sub-mechanisms of neutralization. But agglutination, that's when you're literally like linking together large groups of antigens together or pathogens together, which makes it impossible for them to do whatever effective function they need to do, like invade a cell or something like that. Steric hindrance is when you're physically blocking it from entering a cell or binding to a receptor or something like that. And then that receptor blocker, that's what's shown in this cartoon here. Which is what literally, in the case of this virus, if you have antibodies against the spike proteins on SARS-CoV-2, can no longer bind, can no longer enter. So, other slides I did not put in the review deck, but like with everything else, you do want to kind of understand those antibody effector functions fully. And I think I did put one in here that shows, yeah, I just put one of these in. There's 3 other slides in there that show how these different antibodies binding simply to like an infected cell can attract certain effectors of the immune system. This one is showing attraction of a cytotoxic natural killer cell that is binding to the Fc portion of an antibody and then targeting that cell for destruction. But we also saw examples of complement, where complement is interacting with antibodies, and phagocytosis one as well. And that, so there were some abbreviations: antibody-dependent cell-mediated cytotoxicity, ADCC. There was also an antibody-dependent cell-mediated phagocytosis, ADCP, another slide for that, and a complement-dependent, antibody function as well. Okay, lastly, I know we're getting kind of towards the bottom of the hour. Couple of slides that I pulled to focus on for studying memory, immunological memory, and vaccination. Make sure you kind of understand primary immunity versus secondary immunity, how the differences in the adaptive response. Somebody want to kind of summarize what the main difference is between the secondary and the primary response would be in terms of the adaptive immune response? It's faster, yeah. It's
has a lower activation, so less higher affinity, lower activation, and a stronger response.
Yeah, fewer co-stimulatory signals required, lower activation level, more robust, meaning that those memory B cells, when they differentiate into plasma cells again, they will produce many more antibodies, so stronger in that respect. Stronger, faster, and they've undergone affinity maturation as well, so there'll be tighter, higher affinity binding to those target pathogens. And this box is mostly just reiterating what I said from the lens of the primary immune response. You can just take the inverse of that, and that's what the secondary immune response would be. Memory B cells, these are the cells that are the lucky cells that have survived encounter, the lucky B cells that survived encounter that go on to persist for long periods of time. They don't do anything really except for just wait to bump into the same pathogen again. If they never do that, then they never get reactivated. They don't differentiate back into plasma cells again. But if they do, then they can differentiate back into plasma cells. Maybe a few of them will be retained for additional memory cells, but those plasma cells will be expected to do the same things you do upon first encounter: proliferate and start producing massive amounts of antibody specific against that encounter, that pathogen. Different types of vaccination and immunization, different types of protection. We talked about natural immunity and artificial immunity, and then categories under natural and artificial immunity. So kind of go over these. There are examples of the ones that are less intuitive, but infection is pretty intuitive. Infection is active natural immunity. And vaccination is pretty intuitive as well. Artificial, active artificial immunity. And then if you know what a primary immune response is and a secondary immune response, then you kind of have the basis of how vaccination works. It means when you encounter that again, you're going to get all the benefits of a secondary immune response. Faster, stronger, more robust. I think this is the last practice question. Okay, so you've got a patient recovering from strep pneumo. He had a respiratory infection, and he has long-lasting immunity to that particular bacterium. Which of the following would best explain the basis for the long-term protection? C. Why do you think it's C? Yeah, great. Associate long-term memory of B cells and T cells. lastly, I think this might be the last slide that I've thrown in the deck, last two slides, disorders of the immune system. Particularly, it's all kind of important, but in particular, the superantigen slide is important because we'll hit that so hard in block 3. We'll talk about Staph aureus and other bacteria that produce these superantigens. But nothing too much more elaborate than what we've already talked about, just this constant stimulation of CD4 T cells to produce cytokines that have this amplifying, inappropriate response to this sick superantigen threat. In terms of autoimmune disorders, because we did talk about the way B cells and T cells in particular have to go through down selection and positive selection, autoimmune disorders arise when you have some sort of defect oftentimes in that process. So, causes usually can be a genetic cause, environmental triggers, immune dysregulation oftentimes, but the mechanisms are important to kind of have a grasp of. Don't worry about the examples too much, we'll study these in more detail in block 3. But yeah, that's a whole lot of review. I hope the practice questions helped. Maybe I'll never do those again. I feel like that was a bad thing. Okay. Just trying to make you guys realize that, like, you definitely know this stuff, and you can handle difficult questions. You can do that. But this exam is not a difficult exam. It usually goes really well. And I will try my best to make sure the questions are worded in a really clear way, so there's nothing, like, there are no gotcha questions, there's nothing that's meant to trick you. I just want to know if you're, following the basics of immunology in particular and micro. So, with that, I do not hear about half that, that's okay. Any questions for the group before I let you guys go? Okay, I'll hang around for a while. I do have a student coming by in about 5 minutes, but I can stick around for a while, and then my last student appointment is in 20 minutes from now. Who wants to come out and do this? Thank you guys, and I'll see you.